And this is this story. This is a very this story that I worked on right at the beginning of when I was doing my postdoc and starting my lab. And it's the story of left-right asymmetry. One of the most fundamental things about the body plan of any living thing is what kind of symmetry it has. So we have some things with spherical symmetry and radial symmetry and bilateral symmetry and so on. And separating the body axis into left and right halves is something that happens extremely early. In fact, it's the first step of development. It's getting your bilateral organization right. And in fact, this is why we have bilateral gonadromorphs from butterflies and ants and lobsters, including humans, including birds, which are thought to organize their axis much later on, but I actually think that's false because of data like this. If there are genetic, for example, chromosomal non-disjunctions early on that lead to different coloration or different, in fact, different sex. This is a finch that has a left and right male and female brain. All the divisions tend to be exactly down the middle, because that's one of the first distinctions that cells make when your egg first divides is who's going to be left, who's going to be right. Now, one of the really interesting things about this bilateral symmetry, we are so keyed in to expect and recognize bilateral symmetry that when you take some of the most creative people in the world and you ask them to imagine some kind of horrible, outlandish monster that's going to be as scary and disturbing as possible, they do a reasonable job, but they really can't break out of this bilateral symmetry thing. I mean, look at all this. These are people trying to come up with the scariest, like most bizarre thing they can think of, and all of them are fundamentally bilaterally symmetrical. It is just so alien to us to think about breaking that symmetry in any fundamental way. And yet, biology does this routinely, because even though we are sort of fundamentally have a bilaterally symmetrical body plan, this is that belies a very consistent asymmetry, a consistent left-right asymmetry of things like the heart, the viscera, and in fact, brain lateralization. So early on, we did this, and this is also largely Danny Adams' work, and then a few others. We did some screens, and we figured out that there were some potassium channels and some proton pumps that were really critical for embryos to get their asymmetry right. And when you inhibit those processes during very early development, and you can do that via genetic modification or drugs, this normal laterality where the gut loops to the animal's left and then the heart is on the right, here pointed to the right, and then there's the gallbladder. This can be randomized or reversed by interfering with this process. Now, one of those processes is the fact that at the 4-cell stage, the frog embryos pump twice as many protons out of the right side as they do out of the left. If you inhibit this, either by turning this down or conversely by turning this up, you get the exact same outcome, which is that the organs don't know where to go. They get randomized, and you get heterotaxy. So we studied this process. This is years of work collapsed into a couple of slides. And we found out that the reason that works is because these proton pumps at this very early stage accumulate on the right ventral side. Here they are. And so now we ask, well, how do they get to the right ventral side? And it turns out that there's a biased chiral, and so you can see it's kind of like a right-hand rule, cytoskeletal organizing center that allows maternal pump proteins to sort of ride the microtubules like little rails and all accumulate on the ventral right side. So the details are not important. What this actually does, because once they arrive on the right side, what happens is that they create a voltage gradient that then basically serves as kind of like a little electrophoresis chamber and pulls a morphogen that happens to be serotonin, pulls it all to one side, giving these cells a right identity and giving these cells a left identity. What the important part, what this really is you start off with every cell knowing which direction is rightward, and that's the cytoskeleton pointing rightward, but every cell has the same identical information. And you have to convert that to a positional information of these. So this is a chicken embryo marking off its left side with a nodal left side marker. You have to convert which direction is right into, am I actually sitting on the left or the right of the midline? And the way that works is by this VATPase providing a biased voltage gradient that through this whole body electrophoretic system turns tiny cell level chiral information into animal-wide positional information. Okay, and so I think that's one of the coolest things about this VATPase is that this electrical system shows you how you scale molecular chirality at the level of the cytoskeleton into a large body axis. And so just the last couple of things here. The other thing that we found the VATPase to do, and this is also largely Danny's work, is it's really critical for appendage regeneration. So if you cut off the tadpole tail, normally it will within about a week it will regrow and you get your new tail back. In fact, it's really cool. It grows faster than the normal tail because it knows it has to catch up. By the time the time is done, it has to be as big as the tail would have been by that point. It doesn't just make the same tail. And so what you find is that we can inhibit the VATPase, in which case the tail fails to regenerate, but we can put back a proton pump from yeast, which actually is it has no homology to the VATPase at all, but it has the same bioelectrical effect. And if you do that, you rescue. Here's what it looks like. Instead of in fact, in tadpoles that normally don't regenerate, you can introduce that proton pump, and instead of the nerves here sort of going circumferentially below the surface, they will now come right up to the edge, and they will eventually drive the formation of that whole axis. And so this is really what we're interested in, is this kind of scaling. We want to understand what kind of a cognitive horizon individual cells have, how much memory, how much anticipation, what do they measure, and what happens when it's networked, and how do you scale up these kinds of physiological circuits at the single cell level, things driven by ATP and energy needs and so on, into large scale networks that can process, for example, large scale stress states. So stress is a great marker of cognitive sophistication, because if you tell me what kinds of things you're stressed out about, we can guess what kind of cognitive agent you are. If you can only be stressed by local sugar concentrations, you might be a bacterium. If you can be stressed about global financial markets and what's going to happen hundreds of years from now, you might be a human. How do you scale the kinds of things that stress a particular system? And so we have some models about about evolution scaling up metabolic stress at the single cell level by enabling those stress molecules to basically leak out and get your neighbors to be stressed out about the same things that you're stressed out about. For example, if the cell is in the wrong position, thus raising the plasticity of the whole system, enabling them to get out of your way and being a little more willing to sort of move around. And thus the whole thing looks like it's pursuing a particular large scale structure, such as getting cells where they need to go. So that export of stress together with, I don't really have time, I'm running out of time, so I'll skip most of this, but just this idea of this memory wiping property, that when cells are joined by these electrical synapses, information that belongs to this cell enters the internal milieu of this other cell, and this other cell cannot tell that it's a false memory because it's the exact same information molecules that it uses. So it becomes a memory that's owned by the collective. It wipes the individuality of the cells to some extent and gives rise to collectives that can solve larger problems. So I'll just stop here. And just reiterate the main points, is that there's this scaling of competency from physiology at single cells and metabolics, which are really crucial to getting this whole thing off the ground, to then networks that can solve problems in physiological, anatomical, cognitive, linguistic spaces, and so on. And that bioelectricity is this amazing cognitive glue that turns ancient metabolic states into large-scale kinds of states in morphospace and neurological behavioral space and so on. And the VATPase is one particular piece of hardware that makes it very kind of apparent how this works. So that's super convenient. And I think that by learning to exploit that bioelectrical interface, we can really gain some good applications in, let's say, biomedicine and other things. And so if anybody's interested, all of this is kind of developed in detail in a bunch of papers here. And then I just want to thank the people who did the work. So all the eye stuff was done by Sherry Au and Vaibhav Pai, and Danny Adams did the bioelectrics work that I showed you. Kelly Chang did some of the tail regeneration stuff, and Laura Vandenberg did all the left-right asymmetry stuff. And so I'll thank our funders, of course, the Guy Foundation and others, and Jeremy did all the illustrations. So thank you.